Hello, I'm very happy to be here today to talk about our high-throughput truthing project. My name is Brandon Gallas and I work at the FDA. I spend half my time doing regulatory reviews of medical imaging devices and image-based algorithms. I spend the other half of my time doing research to support those reviews and to improve related regulatory science. I have no conflicts of interest and I'm not endorsing any products. To begin, I want to express my appreciation for all the collaborators that have contributed to this project. We have engaged many through the Alliance for Digital Pathology. I can't do this alone, and I believe it's extremely important to involve experts and the community for true success. I'd like to further acknowledge my HTT Core Collaborators, everybody involved in project management and statistics, the teams that are developing the image viewing and data collection platforms, and the clinical and algorithm subject matter experts. The HTT project is a demonstration project in which we are collecting annotations from pathologists on images to establish biomarker truth. The annotations will then support the validation of an algorithm. We will package these annotations into a submission to the FDA's Medical Device Development Tool Program and pursue qualification. The clinical application, the biomarker, is the evaluation of stromal tumor infiltrating lymphocytes, which are known to be prognostic in breast cancer. In plain language, we have a biomarker. Here you see a figure showing 8 patches of breast cancer tissue. The figure shows the progression of biomarker values from 1% to 90%. The biomarker can be measured by a pathologist or an algorithm. For the pathologist, these measurements take time. They take time for the pathologist to mentally segment different regions and identify the targets within those regions. These measurements require training for pathologists to do these measurements well. And even with training, the measurements from pathologists are noisy versions of the truth. Of course, board certification is required for pathologists to provide clinical reports and practice medicine. On the other hand, algorithms are quite fast at doing these measurements. Of course, they require training to learn the backgrounds and the targets. Once they are trained, they can be quite reproducible. And then, before they can be used in the clinic, they require regulatory permission. Generally speaking, training pathologists and algorithms is quite similar. Essentially, you need examples with truth. Unlike the algorithm, the pathologist can also draw on knowledge from their general training in medicine and biology, and from the literature. To obtain regulatory permission, a developer needs to evaluate the performance of their algorithm. The performance evaluation needs a reliable version of truth, and in this work we are doing truth by pathologist. But we just said that pathologist evaluations are noisy, so we need to reduce that variability, and then we need to account for that variability. How do we reduce pathologist variability? One way is to provide pathologists with additional training and then perhaps test their competence. We also use multiple pathologists per region in order to average over pathologist variability and get a better estimate of truth. In the end, we need to develop statistical methods that account for pathologist variability. All right, let's zoom out from the patch level and look at a whole slide image. Here is a whole slide image of a breast cancer biopsy. These images are quite large, 80 to 100,000 pixels on a side. Images that are 1 gigabyte and larger are common. Because of the size, we reduce the areas of evaluation down to square regions of interest. We select the areas to cover diagnostically important areas of the tissue, areas in the tumor and areas in the tumor margin. We also select other areas that are not diagnostically important in order to test that the algorithm can learn to label them as not related to the tumor. In some sense, we need to do a study to prepare our data collection study. We want to make sure we cover the range of biomarker scores that the algorithm is expected to evaluate. Let's zoom out again and go from whole slide images to patient subgroups. It's time to talk about sampling patients. When preparing a regulatory submission, it's critical to define the patients that the algorithm is intended to evaluate. This is where life gets harder. And convenient image datasets don't always have what we want. Datasets often don't have important metadata about the patients, or they don't adequately sample from important subgroups. For the HTT project, we have identified some important patient subgroups. Unfortunately, we will not likely have the resources to sample and annotate all these subgroups. Therefore, a future algorithm developer will have to fill in the gaps in their regulatory submission. Compared to radiology, truth for pathology can be more challenging. For one thing, pathology images are much larger. That's why we focus on regions of interest. Truth for radiology is often established by pathology. For our biomarker, though, we don't have a confirmatory method, which is why we rely on agreement with multiple pathologists. In pathology, it's sometimes possible to use an alternate tissue stain, a tissue stain that is either more specific or more sensitive for the task at hand. Alternate tissue stains are typically done on adjacent slices of tissue. This carries an obvious shortcoming, as the biomarker can be different in the adjacent slice. It is also possible to restain tissue. Restaining tissue is a complex chemical process. Unfortunately, restaining tissue is risky, as it is possible to damage the tissue. Finally, for both radiology and pathology, outcomes are another version of truth. The challenge is that they are complicated to coordinate and they cost a lot of time, money, and effort. Furthermore, outcomes are several steps removed from the tissue and the biomarker measurement. There are likely many factors beyond the biomarker that affect the outcomes. Not that long ago, we had the opportunity to collect some data at a meeting of the Alliance that was held in conjunction with the annual meeting of the U.S. and Canadian Academy of Pathology. We had 4 workstations, 2 microscope stations, and 2 were digital. We had 64 slides that were split into 8 batches of 8 slides each. And within each slide, we had 10 regions of interest marked. Each batch took approximately 30 minutes. The table here shows a breakout of the data collected by platform. Some of the data was collected at the event and some was collected after through online data collection. All told, we collected nearly 2,400 observations. I want to show you what some of the data looks like. Here is a histogram of biomarker scores for one reader evaluating one case. 6 regions were scored. The other 4 regions were determined to be away from the tumor, and as such, they were not scored. You can see this cluster of scores all the way to the left. This slide yielded low biomarker scores. Many slides yielded low biomarker scores. Here is an example of a slide that yielded moderate biomarker scores. All 10 of the regions were scored, and the highest score was 30. The remaining scores ranged all the way down to 0, demonstrating heterogeneity of the biomarker score within a slide. Lastly is an example of a slide with high biomarker scores. The highest score was 80 and the lowest scores were 20 and below, again showing heterogeneity of biomarker scores within a slide. Alright, now I want to show you the histogram of scores from one reader across all the slides. There are 451 observations here, and you can see how low scores are heavily represented. This may reflect the true distribution of scores that the algorithm will encounter, but we also want to make sure that we adequately sample scores across the whole range. We may want to balance or stratify our sampling method. I want to wrap up this section about the data and talk about the agreement metrics that we will use to characterize scoring differences. There's not enough time to get into detailed math and statistics. I want to simply introduce the metrics with a few figures. Here I'm showing a scatter plot of the paired scores from 2 readers. The plots are scaled with a log base 10 transformation. Otherwise, all the data would be crunched together in the bottom left-hand corner. The circles in this plot are proportional to the number of times specific pairs of scores were observed. Otherwise, multiple observations would lie on top of one another and be invisible. It's arbitrary which reader we put on the x-axis and which we put on the y-axis. We could have put the 2 readers on opposite axes, such as this. Ultimately, we want to put all the readers on both axes and symmetrize this scatter plot. In other words, we will overlay these two plots and put one on top of the other. Alright, here is a scatter plot for 3 pairs of readers evaluating batch 1, scaled by a log base 10 transformation and symmetrized as just described. If we now rotate this figure by 45 degrees, we will get what is often referred to as the Bland-Altman plot. The y-axis now marks the differences of log transformed scores, and the x-axis shows the average of the log transformed scores. Looking at the data this way allows us to identify any trends in the differences as a function of the mean. We summarize the differences with an upper and lower limit of agreement that we calculate from non-standard methods to account for reader and case variability and the correlation structure created by the readers evaluating all the cases. Some of the statisticians in the audience may also be asking about the correlations that arise from multiple observations coming from the same case, the same image. I'll admit that we are still working on methods to account for those correlations. It's a fun and challenging problem. So, what exactly do we do with the Bland-Altman plot and the limits of agreement? We can ask, do differences between an algorithm and the pathologists lie within these limits of agreement? The test statistic will be the average over the squared differences. As my time is nearly up, I want to review some of the important issues that I tried to convey. First of all, in cases when truth is determined by pathologists, we need to appreciate that image evaluation tasks are challenging, and pathologists can be noisy measurement systems. As such, we need to reduce that noise with training and by collecting observations on each region by multiple pathologists. Next, a validation dataset needs to sample the diversity of data expected in clinical use. At a basic level, we need to check that the samples we have cover the range of biomarker values. Additionally, we are trying to find a balance between sampling regions within an image, sampling multiple images within a patient, and sampling patient subgroups. I showed you what our data looks like, and exploring initial pilot data is an important part of preparing a definitive study for a regulatory submission. Regulatory reviewers will ask if your data covers the expected range of biomarker scores and includes important subgroups. I also introduced you to one of our agreement metrics, the Bland-Altman limits of agreement. Whatever metrics we use to characterize agreement, the analysis methods need to account for known sources of variability and data correlations, specifically those from pathologists and images. We refer to such an analysis as a multi-reader, multi-case analysis. The pathologists are the readers and the images are the cases. Additionally, our data samples regions within the images, and these regions can be correlated. We refer to this kind of data as clustered or nested data. There were regrettably many issues that we didn't have time for. At the top of my list would be a deeper dive into the statistical methods just mentioned. My clinical and regulatory colleagues wanted this talk to cover the often overlooked but very important issues of clinical context and clinical use cases. It's not just pixels, pipelines, and Python code. There are patients and doctors, clinical reports, and patient management scenarios. The decisions that doctors make will lead to follow-up procedures and therapies. It is critical to partner with clinical experts so that the algorithm is practically useful. Next, I want to apologize for not spending any time on the Medical Device Development Tool Program. Very briefly, it is a program to qualify tools that can improve and speed the evaluation of medical devices. For our data, we are pursuing a qualification so that the data can be used in a regulatory submission, with little to no discussion and with little to no review. Last, I want to point you to a Device Advice wiki page. The page has many links to relevant guidance documents that you may not be aware of. There are also slide presentations from my colleagues and I that I think you will find useful. With that, I thank you for listening to my talk. We are collecting data to build collaborative relationships, investigate methods and tools, and support the evaluation of artificial intelligence and machine learning. We hope to inform regulatory decision-making, improve submissions, and support and enable interoperability. Thank you.